In this video, we will discuss the effect of this PARP knockdown that will lead to the apoptosis and restore of chemosensitivity of pancreatic cancer cells. So far, we have understood that PARP cause proliferations in the pancreatic cancer cell lines, and now we will discuss if this PARP has been knocked down, then whether it will cause decrease in the cancer cell survival or not. So first, what they did is they checked the apoptosis assay or check the dead cells when the PRP has been silenced, and two different cell lines, that is BXPC3 and CPF-AC1, and compared to the control, we can see that there is a increase in the apoptotic cells for both this siRNA1 and siRNA2. They use two different silencing RNAs to suppress PARP-14 signals in these cell lines. And in both the cell lines, that is BXPC3 and CPF-AC1, they have seen an increase in the apoptotic or dead cells compared to the control whenever this PARP has been silenced. And these things are quantified in the right hand side of the panel A, where we can see that the rate of apoptosis is significantly higher in both the siRNA conditions and in both the cell lines. After that, they check the dose dependent effect of the gemcitabine, which is one of the drugs to target pancreatic cancer cells, and what they are able to see that compared to the control, the apoptotic values are higher for the BXPC3 apoptotic values, or we can say that the amount of gems here we can read it in a way that when the gemcitabine is used, the number of live cells are higher, whereas in the case of the silencing RNAs where PARP has been knocked down or suppressed, we can see the live cells are lower both for siRNA1 and siRNA2. And when they check the IC50 value, it is significantly lower in the case of siRNA compared to the control. And similar things they have observed for CPF-CA1, that is when the silencing RNAs are used, the number of live cells are low with increase in the gemcitabine concentrations, and with also these IC50 values are statistically lower in the case where silencing RNAs are used. And thereafter they check the pro-apoptotic factors are upregulated in those cases or not. So for the BXPC3 they have shown, and they have analyzed these pro-apoptotic factors using Western blot. So for BXPC3 they can see that PARP has been suppressed compared to the control, whereas this cleaved caspase 3 is upregulated compared to the control, and also this cleaved PARP is upregulated in the case of these silencing RNAs. And similar results they have observed for the CPF-AC1, that is for the control they see the lower amount of PARP-14, but cleaved caspase 3 is upregulated compared to the control, and the cleaved PARP is also upregulated. So this indicates that the pro-apoptotic factors are increased, or PARP knockdown triggers apoptosis. And after that, what they did is they prepared cell lines, that is SW1990 cell lines, which is resistance towards gemcitabine. They prepared these cell lines by uh, treating them with gemcitabine concentrations for about 10 months. And this is the control cell lines that they have used, and this is the representative image how this cell lines looks like. And when they check these cell lines against gemcitabine resistance, they can see that cell lines has developed resistance against the gemcitabine compared to the control, and the IC50 values are statistically higher in the gemcitabine resistant cells compared to the control. And when they check the PARP-14 expressions, they see that PARP-14 expressions are increased in the gemcitabine resistant cells, and even the quantitative PCR of PARP-14 shows the similar results. And when in these cell lines, gemcitabine resistant cell lines, when PARP has been knocked down, what they are able to see that this amount of live cells goes down, and they did the dose dependent studies on gemcitabine, and we can see the similar trend, that is compared to the control, that is gemcitabine resistant cells. They can see the number of live cells has lower down when these gemcitabine resistant cells are treated with silencing RNAs against PRP14. And when we check the IC50 value, we can see the IC50 value is also uh, statistically lower compared to its control state. So all this together indicates that this knockdown of PARP14 will trigger or accelerate this apoptosis in the pancreatic cancer cell lines, and simultaneously they also make the cell lines, pancreatic cells, sensitive towards drugs like gemcitabine. I hope you understood how they validate their or how they analyze the PRP14 knockdown cells towards apoptosis and chemosensitivity. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.